Welcome to today's webinar, Food Safety in the CEA Market, brought to you by the Jiffy Group in collaboration with The Ohio State University. I'm Chris Manning. I'm an associate editor with Produce Grower Magazine, and I will be your moderator today. Today's webinar will focus on the following. The market faces a significant problem in ensuring the safety of food solutions, services, and goods. People who will participate in this discussion, hear from experts to get insight into the global context of food safety and the ways in which these factors may affect CEA growers and the use of substrates. We have an expert panel today, so I'm going to run through their bios before I turn it over to them, the people you really want to hear from. First, we have Bart Cron. He is the Director of Quality Health, Environment, and Safety at the Jiffy Group, and he's located in the Netherlands. He has been at Jiffy for 20 years and is an experienced global quality manager with a demonstrated history of working in horticulture. Skilled in ISO 9001, 14001, operations management, international business, quality management, and environmental awareness. Next, we have Janaka Veersura. He's Global Quality Health, Environment, and Safety Manager at the Jiffy Group and located in Sri Lanka. He's been working at Jiffy for 5 years. He's an experienced quality assurance, safety, and health leader in a senior management role with a demonstrated history of working in FMCG and agricultural MNCs. Skilled in quality assurance, consumer safety, product developments on consumer and customer goods of FMCG and agricultural substrate safety and health and factory environments, continuous improvements, Lean Six Sigma applications, world-class manufacturing, Kaizen, and 5S. Next, we have Dana Lucas. She's the segment manager for Indoor Ag/CEA at the Jiffy Group and is located in the United States. She's been working at Jiffy for 1 year. She has a Bachelor of Science degree in Agriculture, Sustainable Food, and Farming. In her role, she's developing new business for Jiffy within the growing Indoor Ag/CEA market. From Ohio State, we have Melanie Lewis-Ivy. Dr. Melanie Lewis-Ivy is an associate professor in the Department of Plant Pathology, Ohio State University, Worcester, with responsibility for research, extension, and teaching in fruit crop diseases. Her research focuses on the development and integration of economical and sustainable practices to reduce plant diseases in small entry fruit and hops. Lewis-Ivy has conducted research in temporal and subtropical climates and placed a strong emphasis on integrated pest management approaches, which can be applied to commercial, home garden, and workplace production settings in any environment. Lewis-Ivy is also the state fresh produce safety specialist with expertise in water quality management. Lastly, we have Sonia Illich. Dr. Illich is an associate professor in human nutrition, Department of Human Sciences at The Ohio State University, and a food safety state specialist. The focus of her research interest is investigating and promoting food safety reduction, prevention of food safety risks among produce growers, food handlers, and populations at risk, including populations with chronic disease. A special interest is microbial safety of fresh fruits and vegetables, and she works with SDEC, Listeria, Salmonella, and others. Dr. Illich investigates transmission and dissemination routes and interventions to prevent and reduce risk of contamination of fresh produce in hydroponic systems, and has authored numerous hydroponic food safety training materials. Without further ado, we're going to first go to Melanie Lewis-Ivy from Ohio State, and we'll start with her and get started with today's webinar.
Okay, thank you, Chris, for that introduction. Today, Dr. Illich and I are going to just briefly talk about some of the key concepts about food safety in controlled environment agriculture. And if we move to the next slide, this is just a very general outline of what we're going to speak about today. And I'm going to start off the presentation, and then when we get to risk assessment, we'll switch over to Dr. Illich. So I'm going to just briefly give a definition of controlled environment agriculture, and then we'll focus on some of the pathogens and microbial hazards that are associated with hydroponic production systems. And then we'll move into the importance of risk assessment, and then briefly talk about prevention and mitigation, or strategies that can be implemented for prevention and mitigation. So I don't actually have a slide for controlled environment agriculture. I just wanted to talk a little bit about what it is. And a very general definition is that controlled environment agriculture, or CEA, is an advanced and intensive form of agriculture where plants are grown within a controlled environment to optimize horticultural practices. And within that definition, when we talk about advanced or an intensive form of agriculture, there are specific levels of control that are included within controlled environment agriculture. So it could be manual control, such as high tunnels, where you manually lift up the sides or open the doors to control the temperature. And it also, manual control has very minimal control in that you're not controlling light, and the only way to control temperature is by managing the doors opening and the sides. And then you can have semi-automated, where you have a bit higher level of technology going into automating the environment. And then you can have fully automated. So there's basically 3 levels of control within controlled environment agriculture. And then the systems that are used within CEA can vary. So a lot of people don't realize that CEA can include soil-based growing. It can also include soilless-based, where you're using substrates such as perlite or coconut coir, or you can have hydroponic, where you're using nutrients as the substrate to deliver your nutrients. And so controlled environment agriculture is very broad. And in the sense that we're going to be talking about it today, is we're going to be focusing on the hydroponic automated systems of CEA. So we can move on to the next slide. So fresh produce causes more foodborne diseases than any other commodity. And in fact, it's around over 50%, around 52% of foodborne illnesses are associated with fresh produce. In terms of foodborne illnesses in general, 1 in 6 Americans will become sick every year from a foodborne illness, and they're not always reported. And so that number is probably an underestimate of the number of illnesses that are caused by foodborne illnesses. We can go to the next slide. So the common pathogens associated with foodborne illnesses in terms of fresh produce include norovirus, salmonella, E. coli, listeria monocytogenes, and hepatitis A. And norovirus is the most common foodborne illnesses associated with all foods. It only requires about less than 10 particles to cause an illness in a healthy person. And you can see here on the slide that I'm showing the minimum infectious doses. And this is assuming that we have a healthy adult. That number can vary depending on the demographic of the person. So people compromised or very young or very old may not need the same infectious dose in order to cause illnesses. The most common pathogen associated with foodborne illnesses is salmonella, as you can see on the slide on the right. And then that's the most common bacterial pathogen. Norovirus is the actual most common pathogen. And then in terms of bacteria, so it's salmonella followed by E. coli and then listeria. And so these are all very commonly associated with fresh produce and are the pathogens that we're interested in studying here within the hydroponic production system. We can move to the next slide. So a lot of the times there's this perception that the risk of contamination within hydroponic systems is less than within the field environment. And in fact, that's not really the case. The risks are similar, but the hazards can vary a little bit. And so when we talk about a field environment, we're often just talking about workers, wildlife, water, and waste management, such as soil amendments like manure. But in the hydroponic system, there are different as well as similar hazards. And so we will talk about workers, that they are a hazard. They can carry the contaminants on their hands and on their shoes as they track it through the greenhouse. Pests are also a problem. The hydroponic environment is not free of pests. Rodents, animals, and birds are risks for contamination of the produce. The surfaces and equipments that is used can have buildup of biofilms, and within these biofilms, you can have Listeria or Salmonella associated with them. So surfaces and equipment are also a risk or a hazard. And then inputs, such as your substrates or your nutrients or the pesticides that you're using, and they're often mixed with water, and then so the inputs can also be a risk. And lastly, the largest risk is your water or your nutrient solution, which can cross-contaminate your product, and then that can lead to a foodborne illness if that product is consumed. So these are the main hazards within the hydroponic system that we focus on, and that our research here at Ohio State is looking at. So the next slide. So who's responsible for food safety within the hydroponic system? And the answer is fairly simple. It's everyone. So everyone through the chain of custody has a responsibility to ensure that the product that they're delivering to the consumer is free of or is not contaminated, has no human pathogen associated with it. And obviously the risk level will never be zero, but we want to work towards that. And in order to work towards that, we need to ensure that everybody is considering food safety within their production system. So that starts off with the suppliers, making sure that you're purchasing product or supplies that are free of contamination, and that you're going to quality vendors when you do so. The farmers or the growers have a responsibility, and that's all the way from the transplants that are being put into the production system to the final product that's being harvested. Distributors, so your storage and handling after it leaves the farm, and then once it gets to retailers or the restaurant or the consumers. So everybody in this case has a responsibility to ensure that the final product that's being consumed is safe and nutritious. Next slide. Okay, so I'm going to pass over now the risk assessment section and our research to Dr. Illich.
So, hi, everybody. It is a real pleasure to be here and talk with you today. So as Melanie mentioned, everybody has the responsibility in food safety. And like in any other step in food production, the risk assessment needs to be conducted in order to understand where these human pathogens can come from, and how they can spread through our production. And the issue with production of fresh produce is that fresh produce does not have subsequent step that is going to eliminate human pathogens. In other words, we don't cook fresh produce. And if there is any contamination at this primary production stage, that contamination is going to be passed on through the value chain and most likely affect the consumer and cause foodborne disease. This is why the prevention is the key. And the only way for us to understand where we can set up these prevention strategies is to conduct thorough risk assessment. Well, in order to prevent risk assessment, we need to understand the hazards. And Melanie mentioned that although hazards are are different than in field production, they're still very much present in the hydroponic production. And unfortunately, we want to say that we're ahead of curve with research, but as it happens all the time, especially in research that is applied like this, we are correcting, we actually use research to correct some myths and confusion about the food safety in hydroponic production. So myth is that hydroponic production is safer, and then we really need to understand those hazards to be able to identify them and set up strategies to prevent them. So the first step is to identify the hazards, and some of those we have, Melanie has already mentioned. Then once the hazards are identified, we're setting up these preventive controls. Well, for that part, we will have to understand what are the strategies that actually work in hydroponic food production. And then finally, it is important that we are transparent and we clearly communicate with our buyers what the risks are. In other words, there's no zero risk that is going to be assumed with any of the inputs or with the product per se. Obviously, the best way to start to analyze the risks is to set up the process flow and build a flow diagram of the product. And this is what growers normally do in the production. And when they do that, they start with they chart the inputs, and they're looking into the safety of their inputs, including substrate that they might be buying from companies like Jiffy. For Jiffy, it is important that you know what your buyer is expecting, and it is important that that you know how to communicate the risks. Can I have a next slide, please, Chris? Thank you. So when we're setting up preventive controls at any point, whether this is in primary production or later on, or within the production of substrate, it is important that any preventive measure is based in evidence. And as I said, research is kind of a little bit scarce in this regard, and we're at infancy. But here at The Ohio State, in Melanie and my lab, we study several important research questions that are going to help us set up the evidence-based effective measures to prevent risks. So for instance, we look for answers to questions: can Listeria pathogens, human pathogens, bacterial human pathogens like Listeria monocytogenes and Salmonella persist in the leafy green production environment in hydroponics, especially in NFT, but also in deep water culture? So in other terms, if the system is contaminated, does that contamination persist? Do the pathogens survive, or do they increase or decrease in numbers? And what are those factors that contribute to their survival and persistence in hydroponic system? How can we trick them into not growing and not surviving in our system? So what are those mitigation practices that are going to reduce the food safety risks? Can I get the next slide, please? Thank you. And this is some of the scientific, the experimental setup that we currently have here at the OSU. We have BSL-2 greenhouses, which means that they are certified to handle human pathogens. And we study Salmonella, Typhimurium, and Listeria monocytogenes to see how long they survive in NFT system if they are contaminated with contaminated water, for instance. And so what we found is that both pathogens survived really well in nutrient solution throughout the hydroponic lettuce growth cycle, both under sporadic conditions or in a situation when we have an extreme conditions like flood or any sort of extreme contamination. And so both pathogens also, unfortunately, accumulated in the rock wool. In this case, in this experiment, we studied rock wool. They accumulated in the rock wool and root matrix, and this actually highlighted the importance of food safety risks that come from the contaminated nutrient solution, but also how substrate can be affected, and how any contamination that exists in the system is going to be remaining actually in the system and not disappearing throughout the leafy green production cycle. This paper has been recently published and it's available. This slide contains the information about the full paper and publication. Can I have another? Next slide, please. Yeah, so we also tested, so now if we do have contamination in the hydroponic system, and we know that these systems are continuous, right? So the new plants are put into the same system as the crops are harvested. We looked into how we can sanitize their surfaces and how we can sanitize the system with or without the plants in them. And as I said, research is correcting myths and confusion. We started testing those sanitizers that are normally in use in greenhouses. And here, in this slide, you can see that we tested Sanidate, Clean Grow, Green Shield, and obviously chlorine, because chlorine is often in use in greenhouses. And some of these sanitizers are not even approved for to be used in food, but they're traditionally used in hydroponics to get rid of different plant pathogens. So here in this slide, you see the effectiveness of sanitizers against salmonella on food grade UV stabilized PVC. So those are that's the material that those NFT channels are produced from. And you can see here that inoculated control has the highest number of contaminants that was not treated. But then you see that when we treat the surface with water or 100 ppm sodium hypochlorite, there is basically no difference. And then you see that the difference is made with higher concentration chlorine, but that concentration destroys this type of plastic, the little tubing, the seals, and causes a lot of damage in the greenhouse to the pumps and also seals. And then you have the stagnant water and all sorts of additional risks. And then a similar situation was with other chlorine-based sanitizers like chlorine dioxide, and in different concentrations. The sanitizers that are not shown in this graph were actually effective, like Sanidate, but Vercon, Clean Grow, and Green Shield are not currently labeled for use in foods, so we could not move them any further in sanitation of these surfaces while we have plants in the system. Can I have the next slide, please? So we see here the deep water culture system that we're also testing. As I said, that only limited number of sanitizers work on surfaces against human pathogens in hydroponic system, and especially even Sanidate that is effective on surfaces has some sort of negative impact on crop yield and crop health. So because of that, we're looking for other ways to remove human pathogens from the system. So we're looking into manipulation of nutrient solution, like lowering pH, etc., and looking into how that affects persistence of Salmonella and Listeria, both in NFT and deep water culture system. And we're working on thorough clean out protocols, because as we know, in majority of these hydroponic systems, there is no clean break almost almost never. They have clean break. So we're looking into effective clean out protocols that are not going to clash with the processes that are existent. And especially we're looking into service sanitation protocols. As you know, there's an algae issue in majority of these systems, and then algae potentially contributes to the problem. Can I have the next slide, please? So to summarize, the foodborne illnesses have severe impact on human health and cannot be taken lightly. So we need to, because the fresh fruits and vegetables is not cooked. We do not have one single technology now known that can eliminate pathogens once they get into the system in this primary production. So the ways to go about excluding human pathogens from the system is to actually prevent their entry at every point, and that really starts with inputs and substrate and seeds, etc. So the food safety training is critical, and awareness needs to exist not only among the producers of crop, but also producers of inputs like Jiffy, and this is what we're doing now. So emphasis on food safety training has to be strong. Maintaining clean environment is critical, because the environment, we assume if the pathogen is present in the environment, it is going to get to the crop. Then the pathogen that is present in the environment can also get into the stored, inappropriately stored inputs, etc., and then spread to throughout the system. Good employee hygiene practices need to be enforced, and this is because the humans are a major factor in transferring human pathogens, not only when they carry human pathogens, but also from the environment. And then risk assessment is critical. We know that in order to prevent any risks, we need to understand them, and we need to foresee them, this is why it is important for hydroponic growers, but also for companies like Jiffy that supply hydroponic growers to choose quality vendors, and then be on the look for the new and effective treatment options, and adopt effective treatment options where available. Can I have the next slide, please? And this kind of concludes our portion of today's talk, and I will let Jiffy now take over from here.
Thank you, Sonia. So we will start now with the Jiffy presentation. So we wanted to highlight food safety in the CEA market, and we have been able to work closely with Melanie and Sonia by getting advice and making sure that we are able to best serve our CEA customers here at Jiffy, especially because we have traditionally been focused in the ornamental industry. And so, of course, food producers have different requirements. Okay, next slide. So just to give a quick introduction on Jiffy. So Jiffy supplies growing media to many different growers all over the world. And we'd like to say that growers can really rely on us to provide a clean, safe, and sustainable and innovative solutions. We offer tons of different products, from pellets, grow bags, to loose substrates, and all of these are considered excellent for maintaining your crop. Next slide, please. So we like to say that, we are respectful, passionate, and reliable, and we do as we say, and we trust people. Next slide. And we also search for continuous improvement and innovations, as well as hoping to reach a common goal with our customers, as well as building long-term relationships, both internally and externally. And we make decisions based on fact, teamwork, and involvement. So a good example would be our work now with Ohio State. Next slide, please. So we are a sustainable, solution-driven, and innovative company, and we care deeply about the environment and are ambitious to help our customers grow and produce food in a sustainable way. And we want to offer tailor-made solutions beyond just our product portfolio. So we're always willing to work with customers and make sure that they get the right solution from our portfolio. And that also goes to us always trying to innovate and make sure that we're accurately serving the market. And again, we just want to make sure that you are served as a customer. Next slide. And I will hand it off now to my colleagues.
Thank you very much, Dana. What is food safety? First time we received the question about food safety, we were surprised. We don't produce food. We produce degradable pots, pellets, preforma plugs, substrates, etc., but no real food. We started to understand that we do have an influence on the safety of the food produced with our products. We started to make risk assessment to find out where possible food safety risks can occur and what we can do to reduce those risks. We have performed a global food safety team and local food safety teams in each production location. These teams are able to implement good food safety practices as well as prerequisite programs which is specific for each products and each location to minimize the food risks. Food safety for me is awareness, building up knowledge, but I realized that we still have gaps in our knowledge and cooperation with the market. Next slide, please. Why does food safety matters? A good question. Everyone has the right to safe food, and that will be a challenge in the future, definitely. Population is growing fast, and it will be a challenge to produce enough food. Developments such as CEA principles will help to mitigate those, but also it brings new challenges in the future. The natural resistance of the crops grown in the way is not that high. In addition, the availability of raw materials to make the right substrates for these crops is under pressure. New raw materials will have to be introduced that may entail more or new food safety risk. Food safety will therefore become increasingly important in the future. Next slide. Thank you. Jiffy Group and quality assurance. Jiffy has a unique quality system. It is called Jiffy Quality Management System, or JQMS. That is one overall global system, which is a combination of management systems supplemented with product certification. For example, we are certified for ISO 9001 and ISO 14001, internationally accepted management system with which the organization can demonstrate that we keep our quality and environmental standards high. In addition, we are proud to be the only company in our branch that has been able to implement ISO 22000 food safety so far. We also have worldwide large number of product certificates that meet the needs of the various markets, such as the organic market like OMRI, FIBL, or the quality certifications of RHP on a lot of our products. Next slide. Food safety and Jiffy's vision. Our vision is to be the innovation and service leader in sustainable plant protein solutions to feed and beautify the world. That means for Jiffy that we have to minimize the risk of food safety. That doesn't mean we can bring them in all situations to zero. For example, human pathogens. It is not always possible, and the next step in the chain of food production has also take some actions to cut off those risks. That means we have to work together. Of course, we already have a good working end-to-end traceability system. And of course, we have a sample schedule for chemical, biological, and physical topics, but that is not guaranteed. For an example, we can't guarantee for human pathogens levels are as zero. We have to do more, and we have to do that with our partners in the food chain, universities, knowledge centers, institutions, etc. But we also have to share our knowledge and doubts. That is the only way to improve in our sector mutually. Next slide, please. Yes, about leadership. Jiffy thoughts leadership or the management commitment. As I said previously, it is impossible for Jiffy or any company like Jiffy to guarantee against human or other pathogens, that is why it is our policy not to recall products in the event that contamination is proven. We really believe that this kind of risk have to be eliminated during most steps in the production of food process. It is not always possible to do this during the production process of our own products. Our raw materials contain natural products like peat, cocoa, bark, etc. that have a huge microbiological live and a strong balance. To destroy all this live and make our products sterile, we open the gate for all types of harmful biological live. We have example of some of greenhouses where the products were sterilized and that led to real disasters. That is why we have to make our products food safe, but it should be step by step, sometimes the edge of our production. As Jiffy, we have taken the lead and started up this kind of discussions with the market, and customers are being explained to explore and to develop methods to improve the food safety in the whole chain of food horticulture. I would like to invite Bart to continue this presentation in the next couple of slides. Over to you, Bart.
Thank you very much, Janneke. Yeah, raw material production and food safety. All our raw materials come from outside, and that's completely the opposite situation on grower side. Our raw materials don't come from controlled environment. It means that nature has an influence on it. Our starting material is not sterile and therefore also has possible food safety risks. We can take measures to reduce those risks, and that's what we do. But food safety risks can never be completely ruled out, not on our side. And the market, so you, must be aware of this and also take measures to further improve food safety. Next sheet, please. One of the most used raw material is peat. And during the production of peat, we already take steps to bring down the risks. Peat is produced on fields of hundreds of hectares. What we do is we create ditches around as barriers against animals, but as you can see, we can't prevent that birds come in. That's not possible. We dry the peat, and that helps to bring down the human pathogen levels, but we can't heat the peat till the human pathogens are destroyed. If we do that, we also destroy the peat itself. Peat also have a very strong natural balance with microbiological life. And this balance that helps to prevent an outbreak of human pathogens. So we have to be careful with the hygienization steps we can make. Next sheet. Another example of an important raw material is coir. Coconuts are growing at the top of tall trees, away from sources of contamination. And after taking out the milk, the nuts itself arrival at Jiffy's. Our drying process is on plates of asphalt and high temperatures above 50 degrees Celsius occur. And that helps. And so we can go further because otherwise we are destroying the physical properties of coir. We cannot go above the 50 degrees. Moisture level will be reduced to below 20%, and that creates a very unfriendly environment for human pathogens. Most of our customers expect that cocoa is a risk because of the tropical circumstances. But we use these tropical temperatures to make it a cleaner product. Next sheet. Another raw material what is being used very often, that's clay. Jiffy's clay is mined in quarries, deep wide grooves that cannot be closed off. The upper layer is removed till a depth that risks like nematodes or human pathogens are not present anymore. As with all of our raw materials, we regularly analyze clay samples to ensure any contamination is kept to a minimum. We always store our raw materials separated from each other to prevent contamination. To explain how our raw materials are being produced, we made a statement with all available type of raw materials to explain what we are doing to minimize the risks, just to be open and to be honest to the market. Next sheet. Storage and production. After the production of the raw materials, which is being done outside, and some steps to hygiene the products are taken, we store the ready-to-use raw materials inside, just to keep more control on food safety. For these logistic processes, we have defined strict hygiene rules for containers, for vessels, for trucks, etc. If possible, we transport it packed and stored it inside till production can start. For some products of Jiffy, we also have steps during our production what brings down the food risks. For example, the Jiffy pellets. We heat the raw material till such temperatures which is enough to destroy human pathogens. The same happened during the production of the Jiffy pots, where temperatures come close till 300 degrees Celsius. But this is not possible for substrates, for example. If we heat that till these temperatures, we will destroy the product. Where possible, we use the hygiene steps what were available, but we have to develop new methods and see where those new methods can be implemented into the production of food. That doesn't mean automatically a Jiffy. Maybe this has to be done later in the process, maybe on grower side. Next sheet. Raw material analysis. Raw materials may be delivered in very large batches by vessel from our raw material harvesting sites to our production facilities. For example, more than 7,000 cubic meters in the case of a shipment of peat. The sample of a human pathogen analysis is 25 grams, which is not representative for a batch of this size. It's not possible to make so many analyses. But also raw materials will come in by truck. We use in high season 3 truckloads a day of core pit. Before we get the analyzers back from the laboratory, we already have used 25 truckloads. We can't wait till we get the analysis result. The value of an analysis is also limited. The contamination will be present. If it's present, it will be spot-wise. And it's a kind of Russian roulette to find it. And even a negative analysis gives no guarantee. But we have a schedule for all our raw materials just to build up data. The input for this sample schedule is coming from our risk analysis, what were made for all our raw materials and for all our production and storage sites, etc., and all the steps we made at Jiffy. We also working together with a provider of tests to find a faster method what can be validated for our raw materials and end products, just to improve our own processes. Next sheet. Yeah, let's work together. A healthy and active soil life is a great strength of our products. These microbes can help prevent outbreaks of human pathogens, even if these are already present. In fact, most of our raw materials are inhospitable to human pathogens. We understand that innovation is still necessary, and we want to continue to develop, working towards common goals based on facts, on teamwork, on involvement, because food safety is not your challenge, because it's not my challenge, it's our challenge. Let's work together. Thank you.
All right, everyone. Thanks again for tuning in today. We are having some issues with video at the current moment, so we apologize for that. But our group, our excellent panelists here, many of whom you just heard on the presentation, will now be unmuting, and we will be taking your questions. If you have them, please submit them into the chat, and we will get to them here in a moment. I want to start with one that Dr. Illich did hit on a little bit in the chat, but I wanted to go back to it. Anne asked, would these apply to aeroponic systems as well? So if Sanjay, Dr. Illich, I don't know if you want to start with that one and kind of expand on it, or if Melanie or anyone else does, but could some of the things we discussed today also apply to aeroponic systems as well?
Yeah, as hi, this is Sonia Illich from the Ohio State University. Question was whether the food safety principles discussed here apply to aeroponic systems, and the answer is yes, they do. Although when it comes to hydroponic farms, every farm is uniquely set up and has its unique set of risks, and that's why we were suggesting the risk assessment. And this is what growers do to really understand the risks in their operations. But the food safety principles that we mentioned do apply to aeroponics as well.
Is there anything anyone would like to add to that before we move on to the next question? All right, I will take that as a no. So this is something I kind of want to hear from everyone on this, but and we've hit on some of this, I understand, but does CEA and just the level of control it can offer growers, does this make food safety any easier for growers to accomplish and zero in on, for lack of a better way of saying it? It's not obviously not necessarily going to be easy, but does CEA offer some advantages in terms of food safety in a practical sense?
Well, I think, as this is Dr. Lewis Ivey at OSU, I think, as we kind of touched on in our short intro, was that there are food safety risks in the field, and there are food safety risks in controlled environments, and some of them overlap and some of them are unique. And the challenges associated with them, vary as well. So I think, just again, reiterating what Dr. Illich said, that, you have to assess the risk within each of your individual units, and that every type of system is going to have unique risks that have to be handled. So, that's a long way for me to say, no, I don't think that it's easier or the risks are less. I just think they're different. And, a lot of the food safety trainings over the past 20 years or more since the development of good agricultural practices for field production have really focused on the field. And we're only now starting to dive into the risks and how to mitigate them in controlled environments.
Is there anyone, I guess, from Jiffy that has something to add to that, or shall we move on?
I think we can move on.
Okay, perfect. So thank you. And again, I apologize to everyone. Again, we're having these video issues. This normally is a little more, I feel like, of a seamless process as we're doing this. We had Anne in the chat asks, and Dr. Illich should answer this one as well, but are there testing kits available for these pathogens to have continuous testing of the CEA system? And I guess, like, can we go a little more into detail about testing? Like, how does that work? How often do you need to test? Just some more detail on the testing part of this.
Yeah, this is Dr. Illich again. Yeah, testing is an interesting an interesting concept, because first of all, it will depend what we're testing. If we're testing the environment, there are test kits that are developed for that purpose, and like a rapid listeria tests or ATP tests, or something that will either test for the presence of the pathogen or a presence for some organic materials that would then indirectly tell us how clean the environment is. So these tests are available, developed for production, for food processing environments, and could potentially be applied in hydroponic environment. But each time that we're testing something, whatever we're testing, whether it's environment or the product, the sampling design is going to determine basically whether you can detect a pathogen or not. So the sampling has to be developed with the outline of each farm uniquely, and anything that we're testing. Obviously, the sampling design has to be statistically sound to actually detect the pathogens, and we know that we also cannot test out human pathogens. So in other words, if we're testing product, hydroponic crops or whatever product is, and we do not find the pathogen, that is not the proof that pathogen is not present, because human pathogen contamination on plants and in environments is sporadic. It is the absence of the pathogen during testing does not necessarily signify that the pathogen is absent. That's why testing is really not our primary way to prevent contamination with human pathogen. We actually, at this point, we think that prevention of contamination at every step is the only way we can with decent certainty say that the pathogen is excluded from the environment. Does that help?
I think that's a great answer. Yeah.
Yeah, and in the US, we have the Food Safety Modernization Act, and it provides some guidelines and standards for source water used to grow crops, particularly source water used to grow crops in the field. And so there are some guidelines for source water testing, but as Dr. Illich mentioned, beyond that, it's just so very complex.
As a follow-up to this, and this is again for the panel, are we still sort of in early days with some of this in terms of how some of these questions, some of these concerns relate to CEA, relate to hydroponics? Like, are we still kind of early on in figuring out how to do this fully and precisely, just based on some of what is available, and maybe some of it coming from being used in other ways before it was used for CEA?
Yeah, yes and no. So we're definitely in early days of really identifying most effective interventions, food safety interventions that it could be easily applied in hydroponics. Because we know that, for instance, production water is one of the could be one of the main sources of human pathogens. To treat the production water that is in immediate and continuous contact with plants is really very different than how we would treat the water in field or any other processing production. So in that sense, we're in early days. But when it comes to testing and testing out human pathogens, there we do have a lot of knowledge about the microorganisms, a lot of knowledge about the statistics of testing, and it is not new that we cannot test out the human pathogens, and that's what actually preceded the development of HACCP and many of the standards that we have now. Then another thing, another problem when we're testing for human pathogens in production is how quickly we can get the results. For again, human pathogens are sporadic and they're often not present in high numbers, in very high cell numbers. So we need to enrich this environment, and that takes time. As a microbiological test, this takes time, and there are still some steps that have not been solved and not been solved with using new technologies. So while the rapid tests technology is evolving and developing daily, we're still not there. We cannot just get into the hydroponic production, pick up few leaves and detect human pathogens. So we can still not do that.
All right, I think we have time for one more question. When we got two similar ones from our viewers today. Daniel and Anne both asked about UV water treatment and the use of UV in being evaluated in hydroponics. So is there any information that is applicable? And what do we know about UV water treatments and how they can be used in these kind of systems?
Yeah, so UV is commonly used in hydroponic systems to treat the recirculating water. And it is important that we treat recirculating water, so it's a good thing. There are a few challenges that we face with UV treatments. The UV light does not penetrate really well through environments that are colored, and we know that the nutrient solution is kind of dark brown. So we have a problem in having the UV light penetrate through that brown colored water. So which means that each drop of the water has to go over the UV light unit in a very thin layer. And this is probably the most effective way that we can sanitize the water, only if the water is going over these units in a very thin layer, which means that each hydroponic system has to have large number of units, especially if they have a high production capacity. So there is an application issue there, and obviously the cost issue of running these units.
And
thanks again. Yeah, go ahead.
I was
Sorry.
just going to quickly say that historically, UV light for hydroponics has been used and been successful for treating plant pathogens. And so, but all of the same things that Dr. Illich just said apply to the plant pathogens as well. So, UV light, has its benefits when it's used properly, just like any other sanitizer. It has to be used, properly for it to be effective.
I'd like to thank everyone again for joining us today, and thanks to our wonderful panel that you heard from. Thanks again to Jiffy for making this all possible and working with us here at Produce Grower. Again, I want to apologize for the issue with the video, but this will be up again on our website, and you can check that out after this has been processed and everything. But thanks again, everyone, for tuning in. Hope to see you again in a webinar in the future. Thanks again to our panel, and have a great rest of your Monday.